These glistening droplets aren't water. They are a dense carpet of sticky, glandular traps. This is the Mexican Pinguicula, a carnivorous plant that has evolved to lure, capture, and digest small insects like fungus gnats to supplement its nutrition. Despite their delicate, flower-like appearance, butterworts are rugged survivors. In their native habitats along the mountain ranges of Mexico, they thrive on isolated limestone cliffs and are incredibly prolific multipliers. Their wild reproduction cycle is highly specialized. Because the plant's stigma forms a protective flap over the anthers, they rely on hummingbirds to bypass these structures and deposit seeds near damp rock walls. But waiting for a pollinator is a risky survival strategy. To endure in harsh, isolated environments, butterworts have evolved to clone themselves asexually, ensuring the colony survives even when seed production fails. In the wild, different species employ 3 distinct methods. Some produce lateral shoots that split from the main rosette. Others send out runners called stolons. A few specialized species even grow fully formed plantlets directly on the tips of their leaves. By artificially triggering this vegetative cloning through a leaf pulling, we can utilize the plant's own regenerative cells to achieve a success rate between 90 and 100%. Harnessing this wild biological adaptation allows a simple houseplant to become a self-sustaining source of carnivorous clones. Step 1. Gather your materials. You need an adult Mexican Pinguicula, a clear-lidded intubation chamber, propagation media, and pure water. Water quality is a strict requirement. Tap water contains dissolved salts and minerals that burn the plant's roots. Use only distilled, reverse osmosis, or clean rainwater. Here are the 2 distinct phases of a Mexican Pinguicula. In summer, the leaves are large and sticky. In winter, it enters a succulent phase, forming a tight, compact rosette of non-carnivorous leaves. Performing the extraction during this succulent phase drastically increases the success rate. These leaves are physically easier to detach from the stem without damaging the plant's core. Now, let's prepare the incubation chamber. For your substrate, use either a 1:2 ratio of vermiculite and perlite or chopped long fiber sphagnum moss. The hydration level must be precise. Moisten the media with pure water until it is damp to the touch, but never soaking wet. Overwatering the media is the primary cause of propagation failure. Saturated soil prevents oxygen flow to the tissue, which leads to immediate rot. Creating this balanced, damp microclimate is a mandatory prerequisite before removing any leaves from the mother plant. With the chamber ready, select an older, healthy leaf located near the outside edge of the rosette. To prevent tearing, gently tease the leaf side to side. Pull downward and away from the stem until it snaps cleanly away from the plant. Inspect the leaf to ensure the white petiole base is intact. These meristematic cells generate new tissue. If left on the stem, regeneration fails. Success depends entirely on preserving that specific point of attachment. Lay the leaf flat on the surface of your damp media. Ensure the white base touches the substrate, but do not bury it. Avoid using any artificial rooting hormones or fungicides. Research shows these chemicals are ineffective for Pinguicula and often result in fatal chemical burns on the leaf tissue. Seal the container with a lid or Ziploc bag to trap high humidity. If you see heavy condensation, leave a tiny gap in the seal to allow for airflow. Place the sealed chamber in a location with bright, indirect light. Avoid direct midday sunlight. A sealed plastic container in the sun will quickly overheat, boiling the vulnerable leaves inside. Your chamber now requires a balance of high humidity, moderate light, and zero chemical interference to trigger the cells to multiply. The transition from a detached leaf to an independent organism typically takes between 2 and 8 weeks. You will soon see tiny green bumps emerging at the white base. These slowly expand into miniature rosettes. Wait to transplant until the new plant has developed 2 to 3 distinct leaves and the original mother leaf has completely withered. Finally, carefully lift the established clone and pot it into a well-draining Mexican Pinguicula soil mix. Mastering this natural mechanism allows you to turn a single plant into an infinite, self-sustaining collection of carnivorous clones.